Photosynthesis. O2, Van Neal. Cyclic photophosphorylation formation ATP. Oxygenic photosynthesis oscillatoria. Stroma in chloroplast of higher plants contains light independent reaction enzyme. PS1 function in cyclic photophosphorylation. Stroma lamellae PS2 and NADP reductase. Kranz anatomy sugarcane. PGA first CO2 fixation producers discovered in algae. Unoxygenic photosynthesis rhodospirillum. O2 evolve manganese and chlorine. Immersion enhancement effect red drop to photosystem operating. Chloroplast higher number of proton simultaneously. Not product of light reaction, lumen of thylakoid. Hatch and Slack pathway, primary CO2 acceptor, NADH. Plastic union, PAP. C4 plant rubisco, photosystem second to cytochrome B6F complex. Non-cyclic electron bundle sheet shell. Cyclic photophosphorylation water. C4 CSNS organ first stable product photosystem 1. Mesophyll cells oxaloacetic acid PAP mesophyll cells. NADP reductase enzyme Lex rubisco. ATP stroma side of membrane. ATP 18 NAD pH2 12 chemoosmosis. ATP is stromal side of membrane. NAD pH2 18 chemoosmosis 12 chlorophyll B membrane proton pump proton gradient ATP synthesis. Xanthophyll yellow green. Carotenoid yellow to yellow orange. Stroma lamellae lax photosystem second and NADP reductase. Chemoosmotic hypothesis electron transport carries step up a proton gradient. Chemoosmotic synthesis of ATP H plus diffuse through ATP synthesis from thylakoid space into stroma. Rubisco uses O2 as substrate. Bundle sheet cell C4 rich in rubisco. Kranz anatomy C4. C4 mechanism of concentration CO2 at a place of rubisco enzyme action. C2 rubisco utilizes O2. Photorespiration starts in chloroplast. CO2 0.03 and 0.04 suboptimum. CO2 increases 0.01%. Rate of photosynthesis increases short term. Photosynthesis and respiration common. Eukaryotes both process occurs in specialized organ. ATP synthesis both relies on chemoosmotic mechanism. Both uses electron transport. Immersion enhance effect and radish. Two photosystems. Photosynthesis pigment located in thylakoid membrane of chloroplast. Transmembrane channel F0 of ATPase. Character of antennae. Photosynthesis more efficient, make a wider range of absorption of incoming light. Dalton center of reaction center is not part of it. Photorespiration. Rubisco binds with O2, phosphoglyceraldehyde and phosphoglycerate formed. Sugar is not synthesized. Scheme. Z scheme. Splitting of water, NADP plus reduction, ATP formation. C4CO2 plus PEP. PEP carboxylase. Biosynthetic phase life uses ATP and NADPH. Strobel and Lamley have photosystem 1. A-granular chloroplast found in bundle sheet cell C4 leaf. Phosphoglycerate 3 carbon. Fructose 6 phosphate 6 carbon. Fructose 1,6 bisphosphate 6 carbon. T.W. Engelmann, Cladophora aerobic bacteria. Most stable carotenoid, most abundant pigment chlorophyll A. Sugar number of क्या है sugar number of carbon mesophyll bundle is 3C 4C 4C Rubisco bundle sheet 5C Rubisco bifunctional enzyme photorespiration neither synthesis of sugar nor of ATP non-cyclic ETS operates photolysis of O2 occurs wheat sugarcane opuntia Calvin cycle limiting factor for photosynthesis is CO2 ATP Calvin cycle of sucrose 24 ATP synthesis chemoosmosis is a membrane a proton gradient ATP synthesis plastocyanin found towards the lumen of thylakoid membrane photosynthesis effects CO2 concentration light intensity light quality C4 primary CO2 fixation occurs cytoplasm of mesophyll cell Z schemes ATP synthesis photosystem second photosystem 1 NADPH water splitting complex photosystem second photochemical reaction light reaction ATP synthesis NADP production during light reaction pH of lumen decreases green plant release O2 engine house photosynthesis pigment in chloroplast embedded thylakoid membrane enzyme dark C4 reaction bundle sheet chloroplast mesophyll chloroplast dark reaction enzyme reaction in to incorporate CO2 plant purify air John engine house cyclic flow of electron occurs stroma lamellus photosystem second photosystem 1 plastoquinone photosynthetic pigment embedded thylakoid first stable product of Calvin cycle 3 PGA pap case mesophyll cell light reaction place in granule photochemical phase ATP and NADPH are produced 5C 3C 5C bundle site cell formation of several layers around vascular bundle impervious to gaseous exchange intercellular space are absent primary CO2 fixation RUBP and PEP carboxylase cyclic phosphorylation ATP formation downhill photosystem second to photosystem 1 produce light reaction O2 NADPH and ATP photosystem second splitting of water injection of electron absorption of photons Maze C3 cycle Kranz anatomy Hatch and Slack pathway PEP NADH formed in glycolysis is oxidized process to form ATP in electron transport system net gain of ATP from one molecule of glucose in glycolysis 2 final hydrogen acceptor ETS of mitochondria is O2 complex iron copper complex fourth without external electron acceptor fermentation 1 glucose complete oxidation yield 36 ATP 22 ATP in fatty acid first degraded to acetyl CoA oxidation NADH during respiration occurs in ETS number of ATP synthesis from 1 turns of Krebs cycle by both substrate level phosphorylation and oxidation phosphorylation 12